Great.
All right. Yes.
And should I get my video going? Hold on. Great. Hi. So you can see me, and hopefully see the screen.
We can see your screen. It's not full screen yet. Yes. All good. Perfect. Thanks.
Okay. Okay. Great. Well, started earlier than expected, but that's great. Just to say thanks very much for the invite. Oh, hang on. My thing is going a bit crazy. 1 second.
So, not full screen. Yes, all good.
Okay, here we go. Let's give this a go. Yeah, thanks so much for the invitation. It's a pleasure to come and speak. I'm talking about some stuff. I guess I've not done a great deal of work on phylogenetics, but this is a sort of topic that came up. So I talk about sequencing error in the context of phylogenetics, and actually slight subtitle being COVID transmission in hospitals. So I guess I got into thinking about sequencing error some time ago, thinking about is you get a genome and lots of reads from that. I guess one thing that's very often done and thought about is how to call SNPs from a genome. So if you've got heterozygosity and you've got some number of reads, you know, what sort of threshold do you put at which to be certain that there is a SNP in the genome? But if you're going beyond that, you might want to perform calculations. So if you find SNPs, you then want to look at different frequencies in the genome. So something I got into quite early was looking at the inference of selection from time-resolved data. So again, you've got data from some system, be that yeast or a viral population, and you fit some model M to your data. So in this case, it's sort of sigmoidal curve. And the thought here is that in order to do this kind of basic model fitting, you need some kind of likelihood function if you're going to do a model fit. It's fairly straightforward. So you could say, well, I'm going to take a binomial function of counts and sort of read depth. But the challenge is in viral populations, you start getting very high read depths. So whereas in your yeast sequencing, you might have a read depth of 100 or 200, some viral populations, you have a read depth of, a million X coverage or something like that. And so at that point, a binomial model seems to give unrealistically large sort of confidence in any particular model. So I was thinking about this and what is the kind of real kind of read depth of a viral population? And one thing I came up with in that is this idea of an effective read depth. So I had a very short paper on this a few years ago, but it was an older idea. And really the concept is that you can take repeat sequencing of a system and repeat samples from a population or repeating of something and look at how much do allele frequencies change. So if you've got a variant at say 50% frequency, is that 50% plus or minus 10%, plus or minus 20%, plus or minus 0.5%? And then sort of, if you like, fit the variance to a binomial model. And that gives you a kind of effective depth. So, if you had perfect sequencing or perfect sampling, what effective depth do you have, so to speak? And it's quite a complex thing. It depends on the read depth in one extent. It depends on the sampling, so any kind of biochemistry, PCR amplification of viral genome and so forth. And it depends on the sort of extent to which a population is homogeneous. So if it's not a well-mixed population, then two samples will be slightly different from each other. So then any kind of mixed model or well-mixed model will be slightly uncertain as a result of that. But the point is, again, I think people know this with next-gen data is that you do have some amount of noise in the data. And I was thinking, well, this also has an implication for a consensus genome, because if you have some, imaginary viral population, and there's a variant that's genuinely sort of close to 50%, with some sort of uncertainty in that, then there's some sort of stochastic change between, being one or the other when you call the consensus, whether it's either side of 50% frequency. So then if we're looking even at consensus data, we could start to see errors in a viral genome sequence potentially. So I got thinking about this and thinking about, can we measure error rates? I'm going to show data. This is 136 people with COVID in sort of hospitals around Cambridgeshire. And so data sets all done by nanopore sequencing. So that's the kind of standard thing. And these are cases where a genome's been sequenced more than once from the same person. And so there's about 300 samples in this plot. And the simple idea is to take the sequence distance on the vertical axis and measure that against time in days on the horizontal axis. And then simply to do a linear regression. And the idea is that the gradient of that gives a rate of within-host evolution. So that is the change in sequence distance over time. That's just a sort of measure of evolution. But then if you take the distance at 0, so this is the sort of intersect of the linear regression, well, that's the kind of distance at 0, which is kind of an error rate, if you want to think of it that way. So I guess we first thought about doing this when we were interested in within-host evolution. So this is data from a PhD student, Casper Lumbey, who was in my lab. And we did this on an influenza B patient, so someone who had flu B for a very long time. And in that case, we were really interested in the rate of evolution in a long-term flu infection. But this time we're interested in what's the rate of error. And so we get a rate of 0.42 substitutions per pair, which suggests that the average viral genome from nanopore sequencing of COVID has about 0.2 errors in it, which seems pretty small. But if you think about a very short period of time, 0.2 errors or 0.4 between two distances, or between two sequences. It's about how much error you would see in 6 days of virus evolution. So what that suggests is that if you're making a kind of global scale phylogeny, this is going to have almost no effect whatsoever. But if you're looking at very short periods of time, this might potentially do something. So here we have some data about COVID transmission, and a measurement from a data set of the number of days between symptoms. So if A infects B, how long after A does B become symptomatic? And sometimes that's negative, but if we take the average, the mean of this distribution gives us the kind of mean time between infections, and that's sort of coincidentally about 6 days. So if we're looking at phylogenetics on the scale of single transmission events, then noise could be doing something. So we can ask, what does it do? And if we think about a kind of likelihood over sort of one transmission and how many sequence changes we'd expect, we can make a sort of Poisson model of how much change there's going to be, what's the SNP distance over one event. And we can see there's a little bit of a difference. So with noise, you can see you occasionally get things with a SNP distance of 3, whereas without noise, you would rule it out. And that's interesting because a lot of people are using kind of SNP cutoffs, certainly in the UK, as a measure of transmission. They say if it's 3 SNPs, that's definitely not transmission. But here it's showing that noise means we maybe need a more inclusive model of sequence change. So moving on from that, I'm going to talk about COVID transmission. This is data from the hospital in Cambridge. And we've got a fairly comprehensive data set. So this is for, it's really for the whole hospital. We've got dates of symptom onset. So that's when people report symptoms. And then also if people are asymptomatic, they are retrospectively asked if they think they did have symptoms. And sometimes they do report having a fever and a sore throat and all these things, even though they came to work. But if we don't have that, then we have a positive test date as a kind of fallback thing. We've then got data saying where all the patients were. So this is hospital records, who was on which ward, and where did the healthcare workers do their shifts. There's also a system to do nanopore sequencing of any samples with sort of reasonably low CT value. So the vast majority of infections have a sample that's gone to a lab and been nanopore sequenced. And in this, we're going to look at data from 5 of the largest ward clusters. So wards have these kind of discrete identities. So going through and looking for a number of cases on a ward and picking the biggest one is what's been done in this particular study. So on to thinking about transmission. As I mentioned, there's a kind of between symptom sort of likelihood, and that can be decomposed into two distributions. One gives the sort of infectious potential relative to the onset of symptoms. And so people can transmit after becoming symptomatic or also before that, over a window of about plus or minus 5 days, according to the study from which we've taken the data. And then after transmission, there's a second distribution, which is the number of days to symptom onset. So we can build a model of a sort of likelihood of, what's the likelihood of transmission, which is a hidden thing on some day, given symptoms between two individuals. So here we've got two distributions. It's a time to transmit from someone who's become symptomatic, followed by a time to become symptomatic from the date of infection. And we can slightly sort of mediate that by location data. So if we know that people were in the same place, we have a kind of prefactor of 1. And if we know they weren't in the same place, we call it 0, because you can't transmit if you're not in the same place as somebody else. And then we allow for kind of probabilistic measures in between. So we get this model, which I've shown at the bottom, which is basically a product of a co-location score and these two distributions that I mentioned previously. And then we can start getting into that as well as the symptom scores. We've also got sequence data. And this is where noise sort of starts to come in. So I've drawn a sort of quasi-phylogenetic tree, albeit with only really 2 individuals in it and some outlier. And what we see is here we're proposing transmission at this time T12. And we've got sequencing of the individual viruses. So this is samples D1 and D2 from the 2 individuals. And we can then write down a kind of likelihood of what's the rate of a Poisson distribution in a kind of phylogenetic likelihood. And here we see it's just a sort of gamma, which is a rate of evolution multiplied by the kind of time between sampling and transmission. So the most recent common ancestor is at transmission, plus this kind of error. So we've already shown that this kind of error term means that we can be a bit looser about the number of SNPs. But there's then a kind of another case to this, which is what happens if the first individual is sampled before the time of transmission. So here we don't really have a most recent common ancestor. If we look at individual 2, that's quite simple because we can say that that individual, the virus has evolved in the time between D1 and D2. So the evolution kind of passes through the transmission event and on. So we've got kind of the same likelihood again. So it's D2 minus D1 times gamma plus this error term. But if we had a SNP in individual 1 that's not seen in individual 2, well, if we take a model of kind of parsimony, we say that SNPs can't really reverse themselves because it's quite easy to gain a SNP because there's 30,000 genome positions, but only one way that SNP can revert. So under a kind of parsimony model, it suggests that variants can't revert. So this would actually be it's sort of impossible to have a SNP in individual 1, but not in individual 2 in this case, unless we allow for error. So allowing for error means that we can see a SNP in the sample D1 arising simply through this sort of non-trivial error. And then it then goes sort of through transmission to D2, and then we don't see the error in D2. So kind of error does seem to do something to our kind of genetic likelihood model. So what we do is we combine this kind of sort of pairwise model of evolution with a pairwise model of symptoms. And we derived a program which we call A to B COVID. And the idea is it's kind of rapid way of telling whether or not individuals might have infected each other. So here's an output from that. The kind of red blocks are points at which people might have infected each other. So on the right-hand side, that's who was in ward X on which day. And the left-hand side sort of shows a kind of cluster down the bottom left-hand side, and then the rest seem to be sort of joined together. So this is a green ward where no one's meant to have COVID, but sort of at least one person has got onto the ward with COVID, and it seems to have spread to lots of patients. And then the sort of epidemiological follow-up can look at these different cases. Here we've got another case. This is from a red ward. So this is a COVID ward where people get put if they have COVID. And here we can see that there are kind of outbreaks, but the outbreaks are quite sporadic. So a few people kind of are infecting each other. In this case, it's healthcare workers getting infected by potentially by each other. So people on the ward are transmitting the virus to each other. So that's our kind of first output. It's a website, and people can check it out and have a go themselves, or stick in your own data and see what comes out. But then the second step has been to go towards the kind of multi-individual model. So here we're really reconstructing the tree of kind of multiple individuals in the ward. And our question in this case is, who is it that infected who? And this has a bit more to it. So the phylogeny becomes a bit more complicated. I mean, it's just really a standard phylogenetic likelihood from, you read people like Felsenstein and so forth, but in a kind of sort of transmission tree with error. And we start off with our pairwise likelihood. And that sort of tells us who might have infected who. And then we look at potential networks. So we try and join these things up in a sort of maximum likelihood way, noting things like shared variants. So when you've only got two individuals, we can account for any sort of set of variants at all, just through error or evolution. But once you start having multiple individuals, you have patterns of transmission that via the presence of shared variants become sort of inviolable. So what we get out of this is these kind of inferred networks. So this is a network of individuals. This is 10 of them. And 9 of them seem to connect up through transmission. But you can see there's some uncertainty. So B0 almost certainly infected B5, B7, and B3. And then kind of might have infected B1, but B5 might have infected B1 as well. So it's a sort of stochastic network of transmission events. And the grey individuals here are healthcare workers, and the white ones are patients. So what we have here is a patient who infects some healthcare workers and maybe another patient, and then the healthcare workers go on and infect each other. So some sort of brief kind of clinical outcomes from this. The first one is that when we look at patients and healthcare workers in hospital, it seems that patients infect each other, and that healthcare workers generally don't infect patients. So that's sort of good news, because we don't want the healthcare workers to infect the patients. But it's sort of still an issue in that patient-to-patient transmission is quite an important thing. Healthcare workers seem to be 50-50, so they sometimes get infected by patients, sometimes by each other. And then we can sort of start looking at these networks. And one of the things we noticed when we do this is to see that some people seem to infect lots of other individuals. So I mentioned this B0 guy who infected at least 3 other people. The network on the right, what you can see is there's a bit of infection going on, but A6 seems to infect quite a lot of people. And then A10 seems to infect quite a lot of people too. So we did an analysis of this, being aware of stuff in the literature about super spreading and reports of that in outbreaks in general. And if we compare a sort of Poisson model where everyone is equally infectious to a model which allows for overdispersion in transmission, so a negative binomial, it's effectively a super spreading model. What we see is there's a significant effect in favor of some kind of super spreading going on. That is, some individuals are transmitting statistically more to others than you'd expect under an equal transmission model. And we can map that. And what we see is 80% of transmission effects, this is our red line, are caused by 20% of individuals. In a way, being a model, it's a kind of continuous line. But what we see is, it's quite interesting in that our model is very close to a report by Adam et al. Earlier in 2020, which was a study in Hong Kong. And so they found that 80% of transmission events were caused by 19% of individuals. And we get that it's 20%. So it's fairly consistent with things that have been found in more generic populations. But here we can show that this is true in a hospitalized population. And so that starts to have implications for the way in which people are cared for. This is another sort of thing we can do from our model, is to go back and infer at what time did these events happen? And I don't know whether this is too small or not, but if you look at when B0 infects B5, B3, and B7, these are the kind of inferred times according to the recipient individual. And what you see is that B0 comes onto the ward, so they've got COVID, but they're put on a green ward by mistake. And really very quickly, they've infected 3 healthcare workers straight away. And that goes on to sort of build a kind of larger transmission network of events. So that tells us that superspreading can happen quite quickly, making a particular challenge for dealing with these kinds of infections in hospital. So some of what we've come to a conclusion as a result of this, one thing we found is that the biggest threat to patients is other patients. And so one of the things that our study points towards is the need really for patients to wear masks. So at the time of the study, everyone had to wear a mask in hospital. The healthcare workers and people on sort of red wards and so forth were all wearing masks. But if you're a standard patient somewhere else, if you're an outpatient coming in from the outside, you have to wear a mask. But as soon as you got admitted, you didn't have to anymore. So it suggests that things to prevent patients transmitting to each other are a good idea. We can go back and look at the individuals who seem to be doing the super spreading. And there's quite a lot of causes. So some are clinical. So they all have underlying conditions and seem to have more severe cases of infection. We can't see that in the CT score, but there are some clinical sort of correlates between them. There's some behavioural reasons. There's the guy who used to wander around the ward and go out for a cigarette every now and then, and he seems to have spread the virus by doing that. And then, of course, environmental causes like ventilation and so forth. We've shown that transmission in hospital, it's hard to stop. So it's still going on. We've got more data from the second wave, and it's pretty bad. But you know, there are things that can be done to stop it. And I suppose most clearly is that integrating lots of different sources of data with sort of phylogenetic and evolutionary techniques gives us an insight into how viral illness is spreading. So my acknowledgements, this is really not as many people who've actually helped in total, but this is quite a reasonable selection of people and a selection of the funders. Just to say thanks very much for listening. Hopefully I can answer any questions. But yeah, thanks very much.
Well, thank you very much, Chris, for the great talk on a very topical subject. And so, okay, we welcome questions either in the chat or speaking up. So, you know, feel free to unmute yourself if you have a Daniel, I think you have a question.
Yeah, Chris, could you just say a little bit more about the beginning? What do you think the main sources of the errors are?
Main sources of error? Well, so I think it comes down to sort of stochastic calling of variants that are at intermediate frequency. So if we look at COVID genomes, most variants are very low frequency. There aren't that many SNPs per genome. But if we imagine that there are a few, sort of, you know, five or 10 above sort of 20%, and it's nanopore sequencing, so it's not the most precise technology for calling variants. So I think what it is it's a kind of stochastic assignment of consensus variants given polymorphism in the population.
Well, I guess what I'm asking about is, you know, you correctly point out that like the binomial model really says that things should be too precise, right? I mean, we've run into this, you know, but like, what is this? What's causing the binomial model to be so bad? Like, why is it so correlated?
Okay, so the previous answer I gave is really about consensus sequences and nanopore. In terms of the within host, so let's say the flu B stuff, we found quite a high sort of within host error. Some of that is to do with the metric we're using is going to pick up really any error there is. I would say what's quite interesting is that if we do replicate sequencing, so we take literally the same pod of stuff and put on two Illumina runs, sequence it to about depth 10,000, then actually a binomial model is pretty good. So it's not the machine, but I think it sort of is some of it's the chemistry. So you get kind of PCR amplification, and that sort of amplifies up errors. And I think something that is probably quite important for viral populations is within host sort of, I won't say within host diversity. What I mean is non-well mixedness, so spatial structure. So it's seen a lot in cystic fibrosis patients who have long-term cystic fibrosis infection. They'll sort of cough up sputum, and if you take a look at the samples, they'll be sort of different to each other. So, I mean, this goes back to some work I did with Katia Corella some while ago, but I think, say, in a viral infection, it's just not well mixed, that viruses go in and infect discrete populations of cells within the airway, within the lungs. And that when people when you take a nasal swab, that's kind of what's in the nose at any point is some sort of stochastic kind of mixture of, you know, when they last sneezed, what happened to come up from what bit. So I'd say it is quite a spatially distributed population. Actually, in the same flu B paper, we see two distinct clades where there's a completely different clade that comes up once in the first 36 samples and looks like it's nonsense. But then kind of afterwards, a bit later, there's a kind of, the infection goes away and comes back. And it seems to be a kind of a resurgence of the second clade. So it's really, so what comes back is halfway between the original sequences and this kind of weird sequence that we saw halfway along and didn't know what it was. So I think quite an important factor is actually the sort of non-well mixedness.
That's super cool, because then, I mean, you know, that noise is letting us peer inside the body and see the structure of the infection in a way that we really couldn't any other way.
Yes, so the amount of noise does. I think the quality of it, you never quite know what you're getting, but I think you can, there might be something that can, well, some different infections I've looked at seem to have different amounts of, you know, differences between samples when done on the same technology. So it's possible that just different kinds of viral infection have different amounts of, you know, genetic diversity, as in not the diversity of a single sample, but the diversity of the within host structure, and that would contribute to it.
Okay, cool. Do we have other questions? Claude, yes, go ahead.
Do we know what is the bottleneck size? I think how many viruses are transmitted from one to the next? I mean, is it something that you can look through the sequences?
So there's some, so I think that's difficult with consensus sequences. It really requires deep sequencing of samples. Nanopore sequencing probably isn't good enough to do that calculation. But some other people have done it. Again, it's a bit tricky. There's lots of technical things with COVID sequencing, and a lot of Illumina data is messy. It seems to be not very many. So sort of in common with flu, I think there's an argument for it to be all kind of respiratory viruses, but it's sort of on average less than 10, I would have said is what I would expect. Katrina Lithgow in Oxford has got a preprint on that, so that would be the best source. That's on medRxiv.
Okay, thanks.
Okay, great. Okay, so I think in the interest of time, we should thank Chris again for his great talk and move on to our last speaker of the session.